Sorafenib, also known as BAY439006, is the first oral multi-kinase inhibitor that targets serine threonine and receptor tyrosine kinases in both the tumor cell and the tumor vasculature. Signals from both outside and within the cell are conveyed to the nucleus through a process known as signal transduction. The RAS protein is a key effector in several signal transduction pathways involved in cellular proliferation, angiogenesis, and cellular death. RAS acts as a switch that transmits external signals through these pathways. In the tumor, one well-known RAS pathway is the protein kinase cascade known as the Mitogen-Activated protein kinase, or MAP kinase pathway. The MAP kinase pathway consists of three kinases, RAF, MEK, and ERK. All transmit upstream membrane signals to the cell nucleus through a growth-regulating signal cascade. In the nucleus, transcription factors are activated, which regulate genes involved in growth and differentiation. A common feature of cancer cells is the deregulation of signaling pathways. While normal cells are highly controlled, cancer cells continuously transmit messages for cellular proliferation. This deregulation can occur through overexpression of upstream growth factor receptors, tyrosine kinases, or through mutations in downstream effectors, such as RAS or RAF. In preclinical models, sorafenib inhibited tumor cell proliferation by targeting the RAF-MEK-ERK pathway at the level of the serine threonine kinase, RAF. In order to grow beyond a minimal size, tumors must induce the growth of new blood vessels to provide themselves with nutrients and oxygen. In preclinical models, sorafenib also exerted an anti-angiogenic effect on the blood vessels that feed cancer cells. The angiogenic process starts with activation of blood vessel cells near the tumor by cell survival growth factors such as VEGF and PDGF. This process leads to blood vessel cell growth, migration, and tube formation. In preclinical studies, sorafenib demonstrated inhibition of angiogenesis through the blockade of the receptor tyrosine kinases VEGFR2, VEGFR3, and PDGFR-beta, and their associated signaling cascades, including the RAF-MEK-ERK pathway, which help to slow or prevent the growth of blood vessels. Sorafenib is the first oral multi-kinase inhibitor that targets serine threonine and receptor tyrosine kinases in both the tumor cell and the tumor vasculature. In preclinical studies, sorafenib targeted members of two classes of kinases known to be involved in both tumor cell proliferation and tumor angiogenesis. Sorafenib targeted upstream receptor tyrosine kinases VEGFR2, VEGFR3, PDGFR-beta, KIT, FLT3, and RET, and downstream serine threonine kinases CRAF and BRAF. Sorafenib inhibited the activity of targets present in the tumor cell, RAF kinase, KIT, FLT3, and RET, and in the tumor vasculature, VEGFR2, VEGFR3, PDGFR-beta, and RAF kinase.